Welcome to Evolution Podcast 9, where we are going to look specifically at how two mathematicians, Hardy and Weinberg, showed us how we could actually measure microevolution occurring in time. So let's get right to it. What Hardy and Weinberg said was that evolution is, just as we were talking in the last podcast, a change in allele frequencies and genotype frequencies, okay, over time in a population, okay? Allele frequencies, how often are we going to see big A in this population? How often are we going to see little a? Genotype frequencies, how often are we going to see big A? How often are we going to see big A, little a, and so on, okay? And then they proved mathematically that a hypothetical population that is affected by only two things: meiosis, the cutting of the chromosomes in half, creation of sex cells, and sexual reproduction. A population affected only by those two things will actually show no change in allele or genotype frequencies over time, okay? And they said that this non-evolving population that is affected only by meiosis and only by sexual reproduction, it also must have the following: a very large population size. Infinite would be nice if we could get it. It must have no gene flow in or out. In other words, no new organisms are allowed to come in, no organisms are allowed to leave. If we're plants, no pollen is allowed to come in from a different location, and so on. No mutations, absolutely no mutations. Mutations, DNA may never change. Mating must be completely random in this population if it is going to remain non-evolving. Okay, no evolution is going to occur from point A to point B. So what that means is that you don't get to pick a mate. Organisms don't get to say, I think you're cute, let's mate. You just basically close your eyes and point, and that's who you mate with. Okay, and finally, that a non-evolving population must have no natural selection acting on it. Everyone is equally fit for their environment. So, holy cow, what populations follow these rules? That only meiosis and sexual reproduction will be affecting the alleles and none of these other things. All these conditions must be met. Of course, the answer is not many. So then why even talk about it? Why talk about some hypothetical, non-evolving population that clearly cannot exist? You can't not have mutations. You can't dictate that. Well, here's why. Hardy and Weinberg said that this hypothetical population would be said to be in Hardy-Weinberg equilibrium. They didn't call it that, but that is what it has been called. And that equilibrium state serves as a model, as a null hypothesis, if you will, a standard of comparison, a mathematical control treatment, if you want to think of it that way, okay? Indeed, of course, natural populations are rarely, if ever, in Hardy-Weinberg equilibrium. However, it is an incredibly useful tool, a useful model to use to measure to find out if evolutionary forces are in fact acting on a population. If we know what a population of these deer or whatever would look like under Hardy-Weinberg conditions, then if they don't look like that, then we can figure that evolutionary change is occurring, and perhaps we can figure out what's happening and in what direction that evolution is going. So, the Hardy-Weinberg theorem involves counting alleles and counting genotypes, okay? So let's think about this. Two alleles, assume we've got two alleles, big B, little b, black fur, white fur, for example. The frequency of the dominant allele, big B, in the Hardy-Weinberg theorem is referred to as little p, okay? The frequency of the recessive allele, little b, is called Q. Okay, so frequency of the dominant allele, how often is big B occurring in the population? That number is called P. How often is little b occurring in the population? That number is called Q. The frequencies of those two alleles, the values P and Q, when you add them, they must add up to 1 or 100%. Okay, 100% or being the value of your whole population. So always P plus Q equals 1. So that is equation 1 in the Hardy-Weinberg theorem. The frequency of the dominant allele plus the frequency of the recessive allele equal 1. Okay. The other part of the Hardy-Weinberg theorem involves counting genotypes. And remember, this is going to be not just disembodied alleles, but actually the genotypes of individual organisms. So, for example, the frequency of the homozygous dominant genotype is determined by squaring the frequency of that dominant allele. So P times P, or P squared. So that's the frequency of big A genotype. How often is that occurring in my population? Is expressed as P squared. Frequency of the homozygous recessive genotype, little a, is expressed as Q squared, little a. How often is that one occurring? And the frequency of heterozygotes, being as there's always two ways you can achieve a heterozygote. You can get the big A from your mom and the little a from your dad, or you can get the little a from your mom and the big A from your dad. Since there are two ways to achieve the same outcome, then the value used to express how often the heterozygotes are popping up in the population is 2pq. So that is the frequency of the heterozygote genotype. Okay? The frequencies of all these individuals must add up to 1, just like with the previous equation. So the other equation in the Hardy-Weinberg theorem is P squared plus 2pq plus Q squared equals 1. Okay? Now, this all may not be making very much sense. Give me a couple minutes and hopefully we'll work that out. So the Hardy-Weinberg formulas in summary. The alleles, P is the frequency of the B allele by itself in the gene pool of your population. Q is the frequency of the recessive allele in the gene pool of your population. How often do each of those occur? And when you add them up, they're going to equal 1. The genotype equation, which is looking at actual genotypes, big A, big B, big A, little a, whatever, okay? The frequency of the dominant genotype, the frequency of the recessive genotype, and the frequency of the heterozygote. P squared plus 2pq plus Q squared equals 1. Okay, so those are our two equations. Okay, so let's put this into practice. Let's use it. Let's say we got a population of 100 cats. We have 84 black cats and 16 white cats. How many do we have then of each genotype? How can we figure that out just by looking at the cats? How many in this population? You got 100 cats sitting in the room in front of you. How many do you have of each genotype? Okay. Well, we can figure out 1 pretty easily because we can know a lot just by looking at it, that white cat, okay? Q squared, which is the frequency of this genotype in the population, we are given here that there are 16 of those. So 16 out of our 100 cats are going to be little b, and we can know that just by looking at those cats because we know that that is the recessive allele and that's information we can easily figure out. We can see that. So 0.16, 16 out of 100, that is the value of Q squared. Okay, so if we want to know the value of Q, how many little b's are in the population, let's take the square root of Q squared. The square root of Q squared is 0.4, so that gives us the value of Q. Okay, so what can we now figure out if we now know the value of Q? We can now easily, because P plus Q equals 1, we ought to be able to pretty easily figure out P. So P is 1 minus Q. If that's true, then P equals 1 minus Q. And so our value for P is 0.6. Okay, so that's how our frequency at which big B occurs in the population. Okay, so if we know that information, then it should be very easy to figure out the genotype frequencies of this population. Okay? But in order to do all of this, we have to assume that this population is in Hardy-Weinberg equilibrium. It may not be, but we have to make that assumption, okay? So if we are assuming that Hardy-Weinberg equilibrium is in place, that it is an infinitely large population, we already know that's not true. There's no mutations, there's no gene flow, and so on and so on and so on. Then P squared is 0.6 times 0.6. And that's 0.36. So 36% of the time, if we were going to pull a cat out of this population of cats, 36% of the time that would be a big B cat. Okay? Then 2PQ, the heterozygote frequency, is 0.48. And remember that's 2 times 0.6 times 0.4. Okay? And that's 0.48. Why the 2? Because there's 2 ways to get that PQ combination. And then Q squared equals 0.16, okay, which we already determined because we could tell that by looking. Now, that gives us this hypothetical, what this population should look like if it is not evolving. Now what we can do is try to, determine and compare over time if this is really what we are seeing. Okay, so assuming Hardy-Weinberg equilibrium, then this is what we expect our cats to look like. This is what we expect the frequency of these genotypes to be. That's our null hypothesis. Okay, so if we actually sample these cats, and what we find is that P squared is 0.74, 2PQ is 0.10, and Q squared is 0.16. Okay, which we know that already. How do we explain that? How do we explain that data? Well, maybe there is some reason that the heterozygote is not fit and that genotype is getting selected against, and this one is being favored. So that's what Hardy-Weinberg can tell us. It can tell us that, whoa, this is not in equilibrium. Why? What's happening? Well, maybe there's something wrong with that genotype. Maybe it's not fit for some reason. And we can start exploring those kinds of questions. Okay? What if we saw that data? Okay? What if we saw P squared equals 0.2 and 2PQ equals 0.64? Well, in this case, maybe there's some heterozygote advantage going on. Maybe this one is now favored, and maybe natural selection, for whatever reason is not favoring that double dominant genotype. Okay? So the Hardy-Weinberg equations are basically tools to determine frequencies of alleles if we know genotypes in a gene pool, and vice versa. If we know at least a little bit of information, we can figure out the whole genetic structure of the population. It is useful because you can figure out what the frequencies should be if the population is in this kind of mythical Hardy-Weinberg equilibrium. And if it's not, then we can kind of start attacking those questions about, well, what's causing this deviation from Hardy-Weinberg then? Why is it proceeding in this path? Why are there more heterozygotes, and so on? So here's a practice problem. PKU. PKU is a disorder that called phenylketonuria. It's a metabolic disorder in which a person cannot metabolize a certain amino acid, and everyone's tested for it when they're born because you can treat it by altering someone's diet and avoiding all the bad symptoms that come with this disorder, mental deterioration and so on. Now, where this has some practical use, for example, in humans, if the evolutionary rate is slow enough, then the population could be close enough to equilibrium that the Hardy-Weinberg equation can actually be used to determine the actual population's genetic structure close enough that it's useful. So, as I said, PKU is a recessive disorder. It can be treated, so we're all tested for it. This disorder occurs at a rate of 1 in 10,000 births. This is real life. 1 in 10,000 babies are born with PKU. So my question to you is, if we know that much information, then what, and we know that this is a very slowly evolving, it doesn't change very much trait. What is the frequency of carriers of this disorder if we assume that the population is in fact pretty close to Hardy-Weinberg equilibrium? So the first thing you need to do, looking at that information, is determine what value have we been given in this problem. The value you were given is that the disorder, which is recessive, okay, occurs at a rate of 1 in 10,000 births, okay? So 1 in 10,000 births are PKU babies. The Hardy-Weinberg value, that is Q squared. That's the frequency of people who have this recessive disease, okay? So if we know that 1 out of 10,000 is Q squared, then what value can we very easily determine from Q squared? That's Q. We just take the square root. And once you know square root of Q squared, excuse me, once you know Q, then you can easily determine P, because we know that 1 minus Q equals P, okay? So once you know the Q and the P, the other values are pretty easily determined. You calculate P squared, and then you calculate 2PQ just by doing the math. So the question was, what's the frequency of carriers in this population? So what is it? Okay, we'll go over it in class, see if you can figure that out. So Hardy-Weinberg can do a couple of things. It can tell us what a population that is not evolving ought to look like so that we can have a standard of comparison. And it can also tell us the genetic structure of a population that is in Hardy-Weinberg, or since that is so very difficult to maintain, at least evolving very, very slowly. Okay, so that's it. Thanks for watching, and I will see you in the next podcast.